Good morning, everyone. Good to be here. Before I start, if you're doing capstone with this class, can you please stay after class? I need to talk to you for 5 minutes before after we finish then before you leave. Okay, so we are still talking about photosynthesis. And by now you're probably very familiar with all of these situations, how the output of the light reactions fits into the carbon reactions, and how it's sort of, at least in paper, a perfect balance between the output of the light reactions and the input of this carbon reaction.
But it reduces both ATP, which provides energy, and also an ATP.
Yes, but that's not really dependent on the regulation. So that's important for it to happen, but how specifically does light regulate the Calvin cycle?
I'm sorry.
Yeah. Don't worry, we have Monday. I think everybody is waiting for Monday. Actually, for today, I'm not putting an alarm tomorrow. Yes, it's available.
So you need light to reduce redoxing, to reduce the enzymes to activate the Calvin cycle.
Yes. So, one more thing. You need two things.
Does it have to do with the conformational change? Or is that later on?
No, that's also important. I mean, it's kind of related, but it's a more specific thing. So, what can you tell me about conformational change?
The sulfur, disulfur.
Yes, and so the conformational change occurs because roughly
It actually, well,
how is light involved in that? In your own words, how is light involved in the conformational change?
well, I'm not really sure on what step the conformational change is.
That's
But like she was saying, you need ATP. So once you have ATP, doesn't that, then it goes to the like you reduce
So
redoxing and then it's after the conformational.
so you mentioned something about the disulfide bridges.
Yeah.
So it's all that, just putting it in a sentence. So you mentioned light and disulfide bridges and conformational change. So without
light, you can't have a conformational. You can't change. You can't break the disulfide bridge and create a conformational change.
Yep, that's it. Okay, so you need light to do many things. So some of the things we talked about is reduce redoxing that only is produced in the presence of electrons provided by the light reactions to reduce a bunch of enzymes. Some of them include how when they are reduced, then you induce a conformational change that activates some of the enzymes. And then also specifically, I just included it here. Sometimes light itself is required for synthesis of those enzymes. So just light can be involved in different ways. One thing, if you kind of remember the exact names, And if you can just describe them, that's it. So what are the different things Rubisco can do? And it can do this and this. And how is this different from this? Yes.
So it can do, it can both work with carbon and it can also work with oxygen as well. So the carbon, it's going to help with carbon fixation, with reducing the carbon into, essentially the end goal is to get it into a 3-carbon molecule, and then it can either go on and become a sugar or just continue through the Calvin cycle. In the other case, if it reacts with oxygen, you're going to end up getting a 2-carbon molecule, which is then going to become toxic, and then I'll have to revert it back in another process to 3-carbon.
Yes. So Rubisco can do 2 different things. If it does what it's supposed to be doing, it is doing a carboxylation reaction. So it adds the CO2 into carbon molecule. Anyway, it gives you 2 of these 3 carbon products. That is what you need to run the candidate sample. If instead of the carboxylation, it takes an oxygen and carries an oxygenation reaction, then what you have is a 3-carbon molecule, because you didn't add anything. You have a 3-carbon molecule and a 2-carbon molecule. And that 2-carbon molecule is basically a waste of energy, because the cycle had to spend energy to recycle this 5-carbon molecule, plus that 2-carbon molecule is toxic for the plant. So we don't want Rubisco to be doing that. So the process, so that is known as photorespiration. So you're using oxygen instead. So the compound, as I said, gives you, well, it's waste of some of the energy and gives you the 2-carbon compound. So it's 2 things gone wrong. So it's toxic. Plant doesn't want it. How does the plant deal with that? Yes.
And then it breaks it back down to H2O and carbon dioxide.
Sorry. What did
Doesn't
you?
it break it back into like H2O and carbon dioxide during photorespiration?
The 2-carbon?
Yeah.
No. Sorry, I just wanted to. So you have this. You have these 2-carbon compounds. That's a long name, but it's quickly. It's toxic. How does the plant get rid of that? That's formed. You have to do something. What does the plant do? Anyway, so let's try to talk this a little bit. So that in the traditional photosynthesis, if things were going usual, the first stable product is this 3-carbon compound. So all of the photosynthesis we have studied until now, it's called C3 photosynthesis. So the first stable product is a C3, a carbon, 3-carbon molecule. In this case, you're producing a 2-carbon molecule. So this is also known as C2 photosynthesis. What can you tell me in general terms about C2 photosynthesis? Or C2 carbon oxidation? Anything related to the first element?
Okay, so I had to look back on this one, but so you take the 2-carbon molecule, you use a salvaging pathway to get it back to the original CO2, so that they can get it back to a 3-carbon butane molecule.
So you're taking the 2-carbon molecule, which is one of the main inputs, and what did you say the output was?
So you're gonna get it back to CO2 so that you can go back to 3-carbon PGA molecule.
So how did you take a 2-carbon molecule, release CO2, and ended up with 3 carbon?
I believe it's the introduction of O2. So they add O2 to the
Yeah.
reaction and it'll react with carbon, get you CO2, and then they take CO2 and put it back at the beginning of the Calvin cycle to get 3 PGA.
Yeah, so in general terms, what you have is you have this carbon molecule that is toxic. You put 2 together, gives you 4 carbon. One of these, you kill 1 carbon, gives you the 3 carbon molecule. That is the 3 carbon that you wanted in here. So that's what the whole process is doing. Let me take 2 of these things. I'll release 1, I don't care, I'll lose 1. But I am recovering part of the carbon, plus I'm getting rid of this toxic thing that I don't want anymore.